Okay, welcome to video number 42 of the Diaries of a Coronavirologist YouTube channel. Today is the 17th of December. We're getting close to Christmas, so I've pulled out my amazing science Christmas sweater, which I found in a box in the lab a few years ago. To move away from that light-hearted note, though, we're up to about 75 million confirmed cases of COVID-19 around the world and a little bit over 1.6 million confirmed deaths. So I've done a lot of videos recently talking about vaccination against the SARS-2 virus that causes COVID-19, and I do want to try and get away from that to talk about some other things, but I've been sucked back in a little bit. So just as an update, I guess, for anyone who's had their head in the sand, the Pfizer-BioNTech vaccine has been approved for emergency usage in the UK and the United States and is being rolled out. People have started to receive injections for this vaccine in both of those countries. The Moderna vaccine, another mRNA vaccine, is just about to get its emergency usage authorization from the FDA here in the United States. So in the coming days, I'm sure we'll start seeing that rolled out in people. Coming up behind those, I think we'll probably have the Johnson & Johnson candidate and the AstraZeneca Oxford University candidate, which will be the next in line to be approved. At least in my sort of Western-centric take on it. There are ones in China and Russia that are being approved as well, but I'm sort of more focused on the Western ones for obvious reasons. So the reason I've come back and been sucked back into talking about vaccines today is because of a paper that was published on bioRxiv on Monday, and it's got the potential to be used for anti-vaccine sentiment, and it already has started to be. So I want to nip things in the bud, put this video here so that if people start raising concerns and questions about this topic, I've got the video available, people have the information available to talk about it and explain why this isn't a concern. And also someone recently asked on one of my videos about this topic, and I briefly talked about it in the previous video, and now there's a paper out there, so that was weirdly timed, and I thought I would therefore tackle this paper. As I say, the paper is up on bioRxiv, which I've talked about many times on the channel, but as a reminder, this is a preprint server. It's not a peer-reviewed journal, it's not peer-reviewed science. It's just science that's been uploaded onto a database. It's as good as putting a PDF up on Twitter for people to read or on Reddit, well, whatever it may be. It's the same kind of thing. This hasn't been peer-reviewed. Were it to be peer-reviewed, this paper would not have fared well, and the only people that would have seen it would have been the editor and the few reviewers, to be very blunt about that fact. The paper is making a very bold claim, saying that SARS coronavirus 2 RNA, so the virus that causes COVID-19, the RNA of that virus can and has integrated into the human genome of patients that were infected by that virus. Now, this is an extraordinary claim to make, and something I'm going to say a lot in this video is that for an extraordinary claim, you need extraordinary evidence. And this paper gets nowhere near producing that. So if you just read the title, this sounds somewhat scary and dangerous. If you look at the paper, it doesn't get anywhere near close to giving data that supports the title. So before we go into talking about the paper, though, I want to talk a little bit about integration of viral genetic material into the human genome, because this actually is something that has and does happen, and it's something I find very fascinating. So a book I read when I was in my undergraduate degree was called Virolution, and this talks a lot about this idea of viral genetic material integrating into the human genome, and that was actually one of the books that really got me interested in viruses to start with, so it's a topic that's quite close to my heart. So we humans at a genetic level are not really all human. Our genome is not just human. There is viral remnant genetic material within the genetic material that we consider our genome. So if you look at it, about 8% of the human genome is made up of this remnant viral DNA. Now this may sound strange, so do bear with me because I find it fascinating and hopefully some other people will as well. In order to explain it, I need to talk about retroviruses. So this is a class of virus in the same way coronaviruses are a class of virus. And the most famous of the retroviruses, I'm sure everyone is aware of, is HIV, the virus that causes AIDS. Retroviruses have an RNA genome, similarly to what coronaviruses have, with some slight differences there. And when they infect a cell, they make that RNA genome into DNA. So they convert their RNA genome into the same structure as our genome in the form of DNA. Now, this is the opposite to what normally happens. In our cells, we use DNA to make RNA, which then makes protein that does everything that makes us a human or makes any life form a life form. So the process of making DNA into RNA is called transcription. And when retroviruses convert their RNA into DNA, this is reverse transcription. Easily named, really. So when a retrovirus infects a cell, so HIV, let's say, infects a cell, it makes its genome into DNA. Then another viral protein is capable of cutting our genome in the cell that it's infected. So it snips, say, imagine a piece of string, cuts that, so you've got 2 bits, and then it ties in its own DNA. Its own viral genome is stitched into the genome of the cell it's infected. That then tricks our cells into thinking that that viral DNA is the cell's own DNA. So it treats it just as it would for any other part of our human genome. It can't tell that that is something foreign at this stage. So because the virus has so well tricked the cell into thinking that the viral DNA is human DNA, that will produce RNA that makes protein that makes more virus particles, and it copies that DNA to make more RNA genomes that make the new virus particles and therefore go on and spread infection. All a virus wants to do is make more particles to spread infection, and that's how retroviruses infect you. Now, importantly, once that viral DNA has integrated into the genome, it is part of that cell for the length of that cell's lifetime. So that DNA will never be removed. As far as the cell is concerned, that is its own DNA at this stage. HIV is a virus that infects cells of the immune system, and in that process, those cells start to die, and that's what leads to AIDS and this acquired immunodeficiency syndrome. However, imagine a scenario in which retroviruses were infecting cells such as sperm cells or egg cells. As I say, once the virus infects those cells and inserts its viral genome into the genome of the cell it's infected, it's there forever. So if a sperm that's been infected with a retrovirus infects an egg, or an egg that's been infected by a retrovirus is fertilized by a sperm, that cell that they produce, its whole genome contains that viral genetic material that's been brought together. So that cell then divides, and eventually you end up with a life form, be it a human or anything else out there. And every single one of the cells in that progeny, that life form that's being produced at the end of all that cell division, they all have that viral genetic material in them because the original one cell had that genetic material. And that's what's happened. This has happened throughout the course of evolutionary history. The billions of years that evolutionary history has been going on, retroviruses have been infecting life forms, and they've got into the kind of cells that drive or that lead to production of progeny, and that's how we've ended up with 8% of our genome having remnant viral genetic material. Now, a lot of it doesn't really do anything. It's not super interesting, but some of it is very interesting. So the placenta can only form because of the integration of a retrovirus gene. So for the placenta, cells need to fuse to form a syncytia, and that is because of a protein that came from a virus. So without viruses, we wouldn't have placental mammals, and therefore by extension, humans wouldn't be here, which I find fascinating. So that's the fun, interesting bit, and I could talk about that for much longer, I'm sure, but to try and keep this video not too long, that's retroviruses. They have the enzymes that can do the reverse transcription to make the RNA into the DNA, and they have the integrase enzyme that allows them to chop up the genome and put their genome in there and trick the cell. Coronaviruses don't have these proteins. Coronaviruses do not have a reverse transcription enzyme. Coronaviruses do not have an integrase enzyme. They can't do that process. And indeed, coronaviruses have been infecting animals on the planet for millions and millions and billions probably of years, and there is no evidence of coronavirus genetic material, any coronavirus genetic material in human genomes or in any animal genome. So there's no evidence in the evolutionary timescale that coronaviruses have ever integrated into the human genome, or into a genome, sorry. And if I have to stress that the fact, evolutionary time is a lot longer than the year we have been dealing with COVID-19. So if this was something that happened, there's a strong possibility we'd have seen some evidence that coronavirus genetic material could integrate into a host genome. The final bit of background I want to just say before I go into talking about the bioRxiv paper is that the human genome is very long. And even though I talk about DNA makes RNA makes protein, only under 2% of the human genome actually makes protein. So when the human genome was fully sequenced, it was thought that pretty much all of it was going to be used to make protein. So it's quite surprising to find that under 2% is what makes the protein. The rest of it was referred to for a period of time as junk DNA. It's not really a term that's used anymore. But the rest of the genome has various other roles. A lot of it is used to make RNA, but that RNA doesn't make protein. So this is referred to as non-coding RNA. Or some of it isn't even coded, it just plays regulatory roles as DNA. So the genome is very complex, there's a lot of stuff I could talk about, but a very small fraction of it actually goes into coding portion, so the part that goes from DNA to make RNA, which makes protein. A big chunk of it makes RNA that has regulatory roles, and a lot of it just stays as DNA. The reason I want to bring this up and stress this point, though, is because if you only ever look at the RNA within a cell and not the DNA, you're only looking at a very small fraction of the genome. You're only going to pick up the coding region, about under 1%, and the non-coding parts, which don't make up the rest of the genome. Not all of the genome goes into making RNA. And that brings me to this paper that was published on bioRxiv. So the main crux of their argument that the SARS genome can be reverse transcribed and integrated into the human genome is the fact that they find RNA that is partially from the virus and partially from a human. So it's a fused RNA, a hybrid, a chimera, any of those words could apply, but they find this hybrid RNA of a viral gene fused to a human gene and make the claim that because they found the RNA, that means that the coronavirus RNA is integrated into the human genome. And that certainly could make sense. If indeed the SARS coronavirus 2 genome is integrating into the human genome, that would produce fused RNAs of the viral genetic material with the host, the human genetic material. So they find these hybrid RNAs by looking at samples from patients. So they've got clinical samples from COVID-19 patients, and they find them predominantly in places where you'd expect the virus to be infecting. So the lungs, for example, have many of these hybrids, whereas the buffy coat taken from blood doesn't. The virus is predominantly in your lungs, not your blood. So that makes sense. Additionally, they find that the most commonly represented viral gene in these hybrids is the N gene. This is the nucleocapsid gene, and that's the most commonly or most abundantly produced viral gene. So that's the one that has the most RNA in a cell it infects. So if indeed the RNA is integrating, you expect to see that the one that's produced the most is the one that's represented the most in the hybrids. So it does sort of track, it sort of makes sense that maybe that has integrated. However, herein lies the problem. To find these hybrids, all they did was RNA sequencing. They've taken all the RNAs in a cell, they've sequenced them, and they found these hybrids. So they're making an argument that the coronavirus genome has been reverse transcribed and has integrated into the human DNA genome without sequencing the DNA genome. They've missed out a whole very important step by not looking at the human genome and just looking at the RNA. And remember as well, I was just saying that if you just look at the RNA, you're missing a lot of genetic information. So they're making a claim for something extraordinary without looking at the very obvious thing of directly looking at the genome to see if there is indeed integrated viral RNA. But they do find those RNA species that are the hybrids of the human and the viral genetic material. So it does still beg the question where those come from, if not from an integrated viral genetic material. Well, there's actually a fairly logical or a reasonable hypothesis that can be made based on how coronaviruses produce the RNAs they need to make protein. So this is a process they use called discontinuous transcription. And it's quite a complicated process. I was planning to explain it on the video. I've drawn pictures and everything, but I've realized it's going to take a long time for me to do it proper justice. So if you're interested, there's various videos on YouTube, or you can read it probably on Wikipedia and places like that. But ultimately, what it boils down to is the fact that when coronaviruses are making RNA to make their proteins within a cell, the protein that makes the RNA jumps around, it moves on the coronavirus genome. It's not a continuous thing, it's discontinuous. So that moving could allow it to accidentally move to a human gene instead of moving back to the coronavirus genome that it needs. And therefore, that's going to fuse a viral gene to a human gene. Now, it's a bit confusing, but it's a very reasonable hypothesis for why you may end up with these fusions. That jumping of the polymerase enzyme could fuse together a human and a viral RNA to give these hybrids. And there's actually very easy experiments to do to see if that's what's happened, because you'd only have RNAs that are expressed and produce proteins. I was talking about those, that small fraction of the genome that actually is coding. It would only be those ones that are part of the fusion, and they could tell that from their sequencing, but they don't discuss that at all. And of course, as I say, the other easy way to discount the alternative hypothesis I've just suggested would be to do the DNA sequencing, and yet they don't do that. So the data is very lacking for integration of the viral RNA into the genome. And again, I'm going to stress the point, to make an extraordinary claim that that has occurred, you need extraordinary evidence, and you need to prove without shadow of a doubt, or well, at least with very strong evidence, that this alternative, very reasonable hypothesis I've just proposed here hasn't occurred. So the bottom line of all that is that they cannot make the claim that coronavirus RNA has been reverse transcribed and integrated into the genome. The data is too lacking, or the data are too lacking for that. However, there could be something very interesting in this if indeed there are these hybrids that are being produced in infection and the virus is fusing its genetic material with human genetic material in the cell, because that could produce proteins that have functions we don't ever think about. There could be something very interesting. We only ever think about the proteins that the coronavirus produces. We don't think about the fact that maybe it could produce protein hybrids with a human cell that might be really important. So there could be interesting data in here, and there could be interesting science to learn from this. But the claim that coronavirus DNA or RNA has been made into DNA and has integrated into the human genome just cannot be made. So that then brings me finally full circle back to why this is frustrating and how it can link to vaccines. People just read the title to this paper and see that coronavirus genomes can reverse transcribe and integrate into the human genome. People are going to make the claim that so too can mRNA vaccines, and this can therefore be used as an anti-vax argument, and it already is being. Honestly, the authors should have thought a lot more about the climate before they put out this incomplete science. Their science does not show the claim they are making in the title, and I'm actually really disappointed to see that this has been put up on bioRxiv. This would get nowhere near publication, so you shouldn't be using bioRxiv for it. Now, I wanted to get this all out there because, as I say, I suspect it's going to continue to be used as an argument for why we shouldn't be vaccinating people with RNA vaccines, because they'll integrate into the genome and who knows what could happen. The paper just doesn't show that. It does not show that this is what's happening. Also, if indeed in a weird world where actually coronavirus genetic material is integrating into the human genome, a virus with a lot more genetic material is going to cause a lot more havoc than a vaccine that has just a small piece of RNA, just one piece of RNA. And you know, a great way to stop the infection with that virus is to get a vaccine. So that's this paper. It was frustrating, as I think I've made clear. But also, with all that said, should the authors continue to work on this and produce significantly improved data that really does convincingly show that the SARS-2 coronavirus genome is integrating into the human genome, then I will recant everything I've just said about this being something that doesn't happen. If they show me good data, I will happily recant everything I've just said. However, that doesn't take away from the frustrations of putting something incomplete out there that will be used in an anti-vax argument. And that's my final thought on this. It's a bit of a heavy video, that one. Thank you if you stuck it out. But there's also interesting stuff around the whole integration of viral genetic material and how that actually drove the evolution of placental mammals and by extension the evolution of humans. So I thought there was some fun stuff in there, and definitely stuff I find fascinating and part of what drove me and encouraged me to be a virologist because I just found that so interesting. So thank you, as I say, for sticking out if you watch the full video. Next week I'll do something a bit lighter. I may even talk about some of my own research because we recently published a paper, and I might talk about that because it does connect to coronaviruses, of course. Anyway, with all that said, thank you for watching. If you found the video interesting, useful, or you enjoyed me ranting, please drop a like for the YouTube metrics. Please drop comments, questions down below if things are unclear or you want further explanations or further discussion on the paper for those who are actually more interested in this topic than maybe the general audience. And please subscribe if you're new around here and you like the videos as well. I've got a reasonable catalogue building up, I guess. Anyway, thanks for watching. As always, please stay safe, wash your hands, wear a mask, and keep calm and carry on. We will get through this pandemic.